helical structures in biomolecules. Coming to helical structures in biomolecules, the most prominent ones, അല്ലെങ്കിൽ helical structures in the ത്രിമൂർത്തികൾ എന്ന് തന്നെ പറയാം. Alpha helix, protein structure ഉള്ളത്, which was discovered in 1951 by Pauling, Corey and Branson. പിന്നെ ഉള്ളത് DNA double helix, discovered by Watson and Crick and published in 1953. പിന്നെ മൂന്നാമത് Collagen triple helix, discovered by Ramachandran and Karta. Coming to alpha helix, അപ്പം ഇവിടെ ഈ സ്ലൈഡിൽ ഞാൻ കാണിച്ചിരിക്കുന്നത് ലെഫ്റ്റ് സൈഡിൽ ഉള്ള ഇമേജ് ഇസ് ദി ഒറിജിനൽ ഇമേജ് ഫ്രം ദ PNAS പേപ്പർ ആസ് ദ പ്രൊപ്പോസ് ദ സ്ട്രക്ചർ ഇനീഷ്യലി. പിന്നെ ബാക്കി കാണുന്നത് ആർ മോഡേൺ ഡേ representations of the alpha helix as such, which will be familiar with most of us in the audience who might be related to biology or biotechnology field as such. പിന്നെ ഉള്ളത് DNA double helix ആണ്. ഇതും മോസ്റ്റ്ലി എല്ലാവർക്കും വളരെ പരിചയമുള്ള ഫിഗേഴ്സ് ആണ്. And this was discovered by Watson and Crick in 1953. So this is the first model or the picture of the first model which was published in the paper in 1953. While this gives the details of the double helix, where you have the bases facing inside and the phosphate backbone on the outside. And then we have the collagen triple helix, which was discovered by Ramachandran and Karta. And what you see is the model which was built by Ramachandran and Karta in the 1970s using wire or using similar to a ball and stick model which we see. So this was the original model built by them and which is right now placed in the Triple Helix Auditorium in CLRI, Chennai. And this is a representation of the triple helix. And I'll come into details of these helical structures soon. So here, before moving forward with the talk, I would first like to define or introduce you to what a helix is. So here, what you're seeing, so all of us are familiar with a circular structure. So now, or even staircases which go straight. So now, instead of a staircase which goes straight, or a spiral staircase ana a helix. So you have a turn, say in a circle, you have a 360-degree rotation. So it is not only a 360-degree rotation, but also a rise. So here, what you see is the depiction of a helix in Wikipedia, where you see a circle. So you start off with a circle and then raise it from one point. That is when you get a helix. And here a helix is defined by the rise or how much rise it makes. So if you consider this as a model of a helix, here, this is a helix which has a longer rise per turn compared to something like this, which is much more twisted. So the helix is defined both by the helical radius, that is the overall radius of this circle shown here. So we know every circle has a radius. So a helix is defined by its radius, and then the rise per turn, or how much does it rise per turn. And not only is there a rise per turn, another definition with respect to helices, especially when it comes to a biomolecule or a repeating unit, which is repeating in a definite pattern, you also have a rise per residue or a rotation per subunit. So now residue is a term which we use in the case of alpha helix. Base is the term which we use in DNA because the component of DNA is the base. And And in collagen, again, it is the residue which comes into picture, because collagen is again made up of amino acids. So for each subunit or the repeating unit, you have a rotation per unit and a rise per unit. And then when you finish one complete turn, you will have a complete rise. So that is the rise of one complete turn. So these are the defining parameters of a helix. Now, what I will take you through is, in order to decipher or find out the structure of the alpha helix, DNA double helix, and collagen helix, what was the information available in the field at the time, and how did these pioneering scientists actually use this basic information and fundamental properties of molecules and arrive at the structures of these prominent molecules in biology. So that is the content of today's talk. I start off with DNA double helix first, because that is the one which is most well understood, or I would say it's simpler to understand than either the alpha helix. And next comes collagen, because that is the most difficult structure of the three to decipher. So that also shows the brilliance of G.N. Ramachandran in deciphering the structure of collagen. So here, this is the double helical structure of DNA, or the spiral staircase. So, a flat ribbon which start either. This ribbon ഞാൻ twist ചെയ്താൽ, that is exactly how so this can be considered a staircase. ഇതിൽ ഇങ്ങനെ ഓരോ lines ഇങ്ങനെ വെച്ചിട്ടാണ് അത് tie up ചെയ്തിട്ടാണ് ഈ ribbon ഉണ്ടാക്കിയിരിക്കുന്നത്. So if I just twist this, that gives you a DNA double helix. And ഞാൻ കൂടുതൽ rotate ചെയ്തുകൊണ്ടിരുന്നാൽ അതിന്റെ rise per turn or pitch കുറഞ്ഞുകൊണ്ടിരിക്കും, or it will be a more coiled or more twisted structure. The first data, or the basis of the main experimental data, is an X-ray diffraction pattern. So this is the famous X-ray diffraction pattern which was obtained for DNA for determining the structure of DNA. And it is the measurement of this pattern by Rosalind Franklin and Morris Wilkins, and followed by the interpretation of the data by Watson and Crick, which led to the discovery of the double helix. So these are the scientists, as I mentioned, who are responsible for the structure of the DNA double helix, as all of us know. So this photograph is said to be among the most beautiful X-ray photographs of any substance ever taken, according to J.D. Barnard. പക്ഷേ ഈ കാണുന്ന X-ray diffraction pattern, or just this black and white image of a few spots, how do you reach to a beautiful DNA double helix is what I will be briefly taking you through today. So at this point of time, what was known, other than this diffraction pattern, what was known was that the number of, first of all, DNA is composed of 4 bases, A, T, G, and C. And earlier, from Chargaff's data, it was known that the molar ratio of A and T were equal, and similarly, G and C were equal. Now, a brief, very layman introduction to diffraction. So now, if you have one atom, or even if you have one small pinhole, if you shine light at the pinhole, you can measure a diffraction from it. So if you have one hole, or now, because we are talking in terms of atoms or biomolecules, so if you consider an atom, and if you shine X-rays, you will get a diffraction pattern. So that is X-ray diffraction. And it is important to use X-rays for diffraction for seeing biomolecules or distances between atoms because the wavelength is related to the distance between the atoms as it is found in molecules. Now, if I have two atoms, as shown here, and if I shine light, or two holes, and if I shine light, I get a diffraction pattern like this. ഇതിപ്പോ നമ്മളൊരു തോർത്ത് അല്ലെങ്കിൽ ചെറിയ തിന്നായൊരു തുണി ഹാൻഡ്കർച്ചീഫ് ഒക്കെ ലൈറ്റിനെ ഇതിൽ കാണിച്ചാൽ you can actually get a diffraction pattern on the wall. Now, if you have a grid of atoms, the diffraction pattern looks something like this. If you have a grid of many more atoms, the diffraction pattern changes. Now, this is what I'm showing here is the diffraction from a crystal. I will also come back to diffraction from a fiber or a helical structure. Before that, what I want to show you is that if you have, say, one structure, so here it is 6 atoms, you get a diffraction pattern like this. If you get many of these, say, within quotes, molecules arranged in a 2-dimensional pattern, you get a diffraction like this. And now, if you have a different arrangement of the atoms, so here, the spacing between the molecules here is smaller than the spacing between the molecules here. What you see is that the spots are much closer together when the distances in the original packing is farther off. So there is a inverse relation between the distances of spot in a diffraction pattern and the distances of spots or distances of molecules or atoms in real space. So that is the relation between real space and a diffraction space. So if you want to interpret a diffraction data, based on the distances in the diffraction data, you can actually find out the distance between the molecules packed in the crystal. So now coming back to the DNA double helix structure and the diffraction pattern, what I'm going to show you is that if you have, say, a layer of lines, which is shown like this, you get a diffraction pattern related to the distances. Now, if these layer of lines is slightly tilted, you see that your diffraction pattern also is different. And here, if you have a set of lines tilted in the opposite direction, the diffraction pattern changes accordingly. Now, if you have a helical structure or a zigzag pattern, as shown in this figure, then what happens is you now get a crossover structure in the diffraction. Now, the next step is that, so this is what I already showed. Now, instead of a zigzag of lines, as shown here, now if you draw a helical pattern in this, you again get a very related X-ray diffraction point. Now, here you see that the distance between these is higher here compared to here where the distance is very less. Now we'll see how the pattern changes. You see that depending on the repeating distance, the cross here changes in shape in the diffraction pattern, and that is similar for a helical structure also. So if you have a helix this way, and you take a diffraction pattern of it, you get a cross structure here. And that exactly is what showed that DNA is a helix. And this is the picture of the X-ray diffraction picture of the DNA. And by now you will be able to correlate how a helix and the diffraction pattern are correlated. Now, depending on whether it is twisted this way or whether it's twisted this way or less, your diffraction pattern will change. the cross will either be broad or less broad. Now, rather than telling vaguely about broad or less broad, what you can measure, or the measurable quantity here, is an angle from the equatorial plane. So this gives an information about the pitch of the helix, or a measure of the degree of rise. So corresponding to this rise, you can get a measurement of this rise. Similarly, you will have a constant radius for the helix, and that is also possible to obtain from this X-ray diffraction pattern. Now, in addition, the repeat distance between the layer lines, or each of these lines, will give you the pitch of the helix. So if you have these two as lines, what was observed in the DNA pattern is that this was 34 angstroms. And that exactly is what they called as the pitch of the DNA double helix, if you know the geometry of the DNA as well. Now, this was a single helix. Now, if it was a double helix, the pattern differs. And what is striking here is that there is a missing line in this point. So if you show two helices here, so if it is a double helix, you see that one of these layer lines goes missing at a periodic spot. So when there are two helices translated by 3/8 of the pitch, you get this one missing. So this is how they found out that, or they deciphered that DNA is a double helix from the X-ray diffraction pattern. Now, in addition to the double helix, you also have these bases stacked, or there are distances between these each sticks. And that distance corresponds to this outermost spot, because a diffraction pattern is inversely related to a real space. So the meridional reflection, or the brightest reflection which you see on top, gives you the distance between bases. So this is how, based on the diffraction pattern, the geometry of DNA helix was deciphered. And once the geometry of DNA helix was deciphered, they actually played around with the AT, GC base shapes. So they made cardboard models of these AT and GC and found the pair for which the sum of these two, or the length of a combination of two bases, was exactly the same. So this turned out to be a combination where G and C are base paired and A and T are base paired. And interestingly, the lengths are same. Not only that, I also showed you that according to Chargaff's rules, the number of G is always equal to C and number of A is equal to T. So then, based on geometry and chemistry of base pairing, along with the X-ray diffraction pattern interpretation, they came up with the structure of the DNA double helix. And this is what they have published in 1953. Based on this, they also mentioned that it has not escaped our notice that when you have a base pairing, this immediately gives you an idea of a possible copying mechanism for the genetic material. So that is how the DNA double helix was discovered. Now, the next story is actually not the next story. Chronologically, the alpha helix was discovered before DNA double helix, and this was by Linus Pauling. Here again, what is important is an X-ray diffraction pattern, which was obtained, one from alpha keratin and also from horse hair. So the diffraction, I don't have an image of that. The diffraction pattern of the alpha helix again gave you the repeat distance, that is the rise per turn and the pitch. And using those values, they tried to match the known chemistry of amino acids. So what was known at that time was the bond geometry of amino acid based on crystal structures of just the amino acid components. So the geometry or the structure of the individual compounds were known, and Linus Pauling, being strong in organic chemistry, was also familiar that the peptide bond is planar because there's a resonance occurring at the bond here. And because of this, there's a partial double bond characteristic here. And this whole set of atoms has to occur in one plane. So, using the concept of planar peptide bonds, and by making sure that, so in order to form a helical repeat pattern, there should be a twisting of these peptides. So instead of considering a straight linear chain of peptides which are linked together, they realized that you have to twist it. So what he did was he actually drew these planar peptides on a sheet of paper and tried to form a helical structure of it by rotating it and finding out which all atoms can form hydrogen bonds. So that helped him to come up with the helical structure of the alpha helix. Earlier models, so people like Kendrew, Perutz, etc., who were working on protein structures, they could not come up with the correct model of the alpha helix because they did not think exactly in terms of the bond geometry, and they missed that the peptide bond was planar. So what this shows is that the chemistry is very important to decipher the structure, and the X-ray diffraction data, which showed the helical repeat, is what helped in understanding the alpha helix. So next, I come to the contribution of Professor G.N. Ramachandran of the triple helix of collagen. So just like alpha helix, so alpha helix was discovered in 1951, DNA double helix in 1953, and by 1955, G.N. Ramachandran could propose the triple helix structure for collagen. So here, what exactly was the experimental data? Again, there was a diffraction image in the picture. So here he obtained the diffraction data from kangaroo tail tendons, which is very high in collagen. And he used that as the sample and collected the X-ray diffraction pattern, which gave the rise per residue, which is 2.9 angstroms, and the diameter of the helix, which is from the equatorial spots. And another thing which was known about collagen was the amino acid composition. It had a repetitive amino acid composition where every fourth residue was a glycine. And it also had a high propensity or high abundance of proline and also hydroxyproline. So both glycine and proline, for those who are familiar with biochemistry of amino acids, will know that they are special. So glycine is the smallest amino acid, which means that there are some positions in a protein structure where only glycine can occupy. On the other hand, proline and even hydroxyproline, which is a derivative of proline, it has an extra OH group. So proline is also special because it is an amino acid in the sense that the side chain goes and forms bond with the NH2 group again, and it's an amino acid, and which means that the conformations which it can take are restricted, And not only that, the availability of a hydrogen for hydrogen bonding is not there for prolines. So this hydrogen attached to NH is unavailable in a proline for hydrogen bonding. So this is a very interesting fact about how Prasad and Ramachandran came up with the idea of a triple helix structure. So they say that in one of his talks, he mentioned that it is based on, he derived the idea from astronomy. So the moon, when it rotates, it always revolves around the earth, but then it always presents the same side to the earth. So that is exactly what he was reminded of when he thought of glycine residues, because you have glycine coming in a repetitive pattern. And this fact made him think that glycine should always face the center of the triple helix, because glycine is a residue which requires least space among the 20 amino acids. So these glycine should always face the center of the triple helix, and that is how he came up with this structure. I know that this is very difficult to, so this is the figure from his original paper, which is difficult to understand, and all these are the distances which is being shown here. And this is a much modern representation, so to say, a space-filled model of the atoms of the triple helix. And what you see here, so you see each of these strands in a different color, and each of these form, so here I've written that it is 3 lefts and a right. So here, each of these helices is a left-handed helix. So now, one feature which I did not mention about helices is the handedness. So you have either a left-handed helix or a right-handed helix. So now if you look at this structure here, if you, so to determine the directionality or the left-handed or right-handed, it depends on, so you can figure this out based on the the direction of your thumb and the direction in which your finger goes. So you can have either left-handed or right-handed structures. So here you see that this magenta, red, and yellow helices, they are all following a right-handed structure. So if you are thinking the upward direction is the direction of the thumb, you can see that the helices are coiling in a right direction, right-handed direction. So this is my right hand. Now, if you look at an individual orange helix, which you see here, I know that in this figure it's a slightly smaller version. So here, this twist of the helix. So if you look at only the orange version, this is actually a left-handed helix. So just this orange portion is a left-handed helix. So that is how there are 3 left-handed helices coiling around to form a bigger right-handed helix. So this is the structure of collagen, where you have a repeat of glycine, proline, hydroxyproline, again glycine, proline, hydroxyproline, and it goes ahead. So wherever the glycine faces, the glycine will be facing the inside of the triple helix, and the prolines will be facing outside. So I mentioned to you that the amino group, so here the nitrogen is shown in blue. The amino group does not have a hydrogen for forming a hydrogen bond and forming a stable structure. So what holds the 3 helices together in a collagen structure, as proposed by Ramachandran, was the hydrogen bonding between one chain and the next chain. That is, between 2 different colors, which are shown here. So that hydrogen bonding holds the chains together in a collagen triple helix. And then there are also in other hydrogen bonds which are water mediated, which again is the importance of hydroxyproline. So this is exactly where the origin of the Ramachandran plot comes in. So when he proposed this structure, and when he proposed that there can be an interchain hydrogen bond between NH and C double bond O, between the two chains of collagen, that is where there was an opposition saying that the geometry does not allow atoms to come as close together as was proposed in the collagen structure, and they said that his structure is wrong. So that is where he went into doing an exhaustive analysis or exhaustive cataloging of all possible combinations of all possible rotations of phi-psi angles, which can be accommodated in a structure. And that is how the Ramachandran plot, as all of us know, came into picture. And in the Ramachandran plot, GNR's collagen triple helix actually occupies a very special position because, as I just mentioned, collagen is a protein which has a very special or unique amino acid composition, which is mainly composed of proline and glycine. So if you consider this as a general Ramachandran plot, which is derived with alanine, by doing alanine dipeptides, and this is applicable for all other 20 amino acids except glycine and proline, where you see that glycine, especially being a non-chiral amino acid, and because it has less steric hindrance because of the absence of the R group, forms a mirror symmetric Ramachandran plot, and it has more allowed regions in the Ramachandran plot. Whereas proline, the other prominent residue in collagen, forms a very restricted region. So there's a restricted place which it can take up, or a restricted conformation which it can take up in the Ramachandran plot. And now a residue before proline, which is called as pre-proline, actually again has a special preference for or phi-psi. And another speciality of a pre-proline is a pre-proline residue, or a residue or an amino acid which comes just before proline can potentially form a cis peptide bond also. So many of the people who were pursuing the structure of collagen considered the pre-proline structures to take up possibly a cis peptide bond, and hence they could not arrive at the structure of collagen. But Ramachandran correctly placed this as a trans peptide bond, and hence he could get a thin extended structure which matched with the X-ray diffraction pattern obtained experimentally for collagen, that is with a Rice-per residue of 2.9 angstrom, which is a very extended structure. Extended, or so this is a thin, each of these individual strands is a thin helix compared to that of alpha helix. Yeah, so with that, I have described sort of the experimental context, and one of the things which I find most interesting with this story of helices is that here all these scientists who are pioneers in the field have come up with this astounding structures which were totally unknown earlier, and all based on logical reasoning. And so what they had was distance measurements, which the X-ray diffraction pattern gave them. And then they made maximum utilization of the chemistry of the individual components which was available at that time. And it's essentially like putting together a puzzle that these structures were deciphered. So that is what I find most beautiful in the story of helices. And we should all be proud that Professor G.N. Ramachandran and his colleagues deciphered the structure of the collagen triple helix, which among these three structures, I find as the most difficult one to solve. And not only that, it is also the structure of the most abundant protein, which is present in animals.